Welcome to part 16 of this video on the immune system. Okay, so we're now going to continue on with the story of the adaptive immune system. So remember, the point that we've got up to so far is there is this pathogen infecting our tissue, and what's going to happen is an antigen-presenting cell, which is either a dendritic cell or a macrophage, most likely a dendritic cell, is going to chop up the antigens of that pathogen and present fragments of those antigens on MHC protein complexes. And remember, if the pathogen is intracellular, then its peptide fragments will be presented on MHC class 1 protein complexes, whereas if the pathogen was extracellular, the peptide fragments are going to be presented on MHC class 2 protein complexes. Okay, so we now want to continue the story on from there. So what's going to happen is the antigen-presenting cell is going to now go and present these antigen fragments to T cells, and it's going to try and find T cells that have the correct Tcell receptor that they can be activated by one of these antigen fragments that's on the surface of the antigen-presenting cell. And remember, the antigen-presenting cell in reality will have a huge number of different MHC complexes on its surface, which will have very different fragments of potentially different antigens of that pathogen on its surface. So one pathogen will have a huge number of different antigens, and all of those antigens will have a huge number of different antigen fragments, and you'll have this great plethora of antigen fragments on the surface of the antigen presenting cell, either on MHC class 1 or MHC class 2 complexes, depending on whether the pathogen was intracellular or extracellular. Okay, so the antigen presenting cell just needs to find one T cell clone that can be activated by one of those antigen fragments, and that's the story that we're now going to follow. We're going to follow the story of one of the antigen fragments that we managed to find a T cell which has a T cell receptor that can be activated by the MHC complex with that antigen fragment in it. Okay, so the antigen presenting cell then, it's going to need to go and excuse me, it's going to need to go and actually find the T cells. Now we've seen where the T cells are going to be present. They're going to be present in lymph nodes, and this is actually the way that the antigen presenting cell will get from the peripheral tissue where the infection was present to the T cells. It's going to go into the lymphatic vessels that drain that tissue, and then it will gradually drain into bigger and bigger lymphatic vessels, and then eventually it will get into a lymph node. And then in that lymph node, it may well find a T cell with the correct T cell receptor. So just going back to my picture here, so if we go for this picture, the antigen presenting cell will drain into the lymphatic system, and then it will move with the lymph, and let me just straighten this up. It will move with the lymph to a lymph node, and then what's going to have to happen to this antigen presenting cell is it's going to have to go from the marginal sinus to the medullary sinus, and to get through there it's going to have to go for a huge number of T lymphocytes, and therefore it's going to come in contact with a huge number of T lymphocytes. And the idea now is that antigen presenting cell, it's coated in fragments of antigens from our pathogen. Hopefully it's now going to come in contact with a T cell that will be able to respond to one of those antigen fragments, and that's the story that I now want to follow. Let's suppose it does manage to find one of these T cell clones that has a T cell receptor which can bind to and be activated by one of these MHC complexes with a peptide fragment from an antigen of the pathogen in it, and we want to follow the activation process now. And actually, this is how important recirculation of the T lymphocytes is. So this stresses the importance of recirculation, because we do need to continually be moving our T cells around so that they go into all the different lymph nodes of the body to make sure that the different T cell clones aren't just clustered. You know, they're not just staying in one lymph node. They need to move around the different lymph nodes because we're potentially going to need them in different areas of the body. So that's why T cell recirculation is extremely important, to make sure that the antigen presenting cells can actually find the correct T cell clones, and they're not in some lymph node that the antigen presenting cell from that area of tissue can't really find. Okay, So let's now go over the process of T-cell activation. Right, so let me just straighten this up. Okay, so initially what I want to do is just draw a very basic picture of what's going to happen, namely that the MHC complex is going to bind to the T-cell receptor, and either CD4 or CD8, remember the co-receptor for binding to the MHC complex. Then what I want to do is discuss the signaling pathway that this is going to activate in the T-cell to some degree, and will follow how the T-cell is actually going to be activated by the antigen-presenting cell. Okay, so let me draw this on here. So we'll draw our antigen-presenting cell here, and again we'll just assume that it's a macrophage here because it makes the picture somewhat simpler. So here, this can be the nucleus of our macrophage here, and let's say it's got an MHC complex on its surface. And here again we will be staying general, so I'm not going to say whether this is specifically MHC class 1 or MHC class 2. Remember, if we're dealing with an intracellular pathogen, it will be MHC class 1. If we're dealing with an extracellular pathogen, it will be MHC class 2. Okay, so this could be either MHC class 1 or MHC class 2. So at the moment, the cell-mediated and the humoral adaptive immune responses are completely the same, and therefore all of what we're discussing applies for both of them. In a moment, after we've discussed T-cell activation, I will diverge the two pathways. We'll discuss cell-mediated adaptive immune response, and then we'll go on to the humoral adaptive immune response. But for now, they're both identical, so we're going to discuss them both at the same time. Okay, so this is going to be either MHC class 1 or MHC class 2. MHC class 1 is for the cell-mediated adaptive immune response, MHC class 2 is for the humoral adaptive immune response. So let's just colour our MHC in here, in orange. Okay, there we go. And let's now put a little peptide fragment in the peptide binding groove of the MHC that I've left here. So here, this is going to be the peptide fragment, and remember, if we're talking about an MHC class 1 molecule, then that peptide fragment will be between 8 and 10 amino acids, whereas if we're talking about an MHC class 2 protein complex, that peptide fragment will be between 12 and 20 amino acids. Okay, so there is our MHC complex with the peptide fragment in on our antigen presenting cell here. And we're now going to suppose that this specific peptide fragment of one of the antigens of the pathogen on this MHC complex we've actually managed to find a T-cell with a T-cell receptor that can actually bind to this. And remember, you have a huge number of different T-cell clones. There are a huge number of different T-cell clones, and those different T-cell clones will have different T-cell receptors on their surface. So you'll have to search and search and search for an appropriate T-cell clone that has a T-cell receptor on its surface which can bind to one of these MHC complexes with a peptide fragment from the pathogen on it. Okay, so let's now begin drawing the T-cell here. So this can be our T-cell here. I'll just give it a nucleus as well. So T-cells have very large nuclei. They're taking up most of the cytoplasm when they're in the inactive state, which indeed this one will be. This is a mature, naive T-cell, the type that we've seen now the production of, and which will just be recirculating continuously between the lymphatic system and the circulatory system. Okay, so on its surface it's now going to have a T-cell receptor that's going to be binding to this MHC protein complex with this peptide fragment in it. So here, this is going to represent its T-cell receptor. Okay, so I'll colour this in red here. So this is the T-cell receptor, and I'll also just label up the mature naive T-cell as being such. Okay, so this cell here, this is the mature but naive T-cell because it's never been activated so far by antigen. It's never seen its antigen that it's targeted against so far. Right, so next up what I want to discuss is the fact that we're going to have a co-receptor also binding to the MHC class 1 or MHC class 2. Now remember, if you have a T-cell clone that has a T-cell receptor which combines to an MHC class 1 protein complex, then the co-receptor for that will be CD8, whereas if you have a T-cell receptor that combines to MHC class 2, then the co-receptor for that will be CD4. And there are those two populations of T cells, the CD8 positive T cells, which are going to be part of the cell-mediated adaptive immune response and are also known as cytotoxic T cells, and the CD4 positive T cells, which are going to be part of the humoral adaptive immune response and which are also called the helper T cells. So I'll draw the co-receptor here. So if this is an MHC class 1 complex, then we'll be wanting to activate the cell-mediated adaptive immune response, and therefore this will be a CD8 positive T cell that will be activating a cytotoxic T cell, whereas if it's MHC class 2, then we'll be wanting to activate the humoral adaptive immune response because it will imply that the pathogen is extracellular, and therefore we'll be wanting to activate CD4 positive or helper T cells. So this will be CD8 if we're talking about MHC class 1, or CD4 if we're talking about MHC class 2. So I'll just colour this molecule in here. And remember that each mature naive T cell will only be CD8 or CD4 positive. There aren't any double positive mature naive T cells. Okay, there we go. Right, so this is what's going to form this interaction here between an MHC complex with a peptide fragment in, and then the T-cell receptor and the co-receptor. And this is now going to lead to a signal being propagated intracellularly inside the T-cell. And what I now want to study is this signal in more detail. But before we do study this signal in a little bit more detail, let me just give you the name for this. This is known as signal 1 for the activation of T-cells. So the formation of this sort of a complex here is going to deliver signal 1 to the mature naive T cell. And what I do want to do now is just study this in a little bit more detail. So I want to study the T-cell receptor in a little bit more detail because it actually forms a complex with an important protein complex known as CD3. And I want to discuss the beginning of the intracellular signaling cascade that is actually caused inside the mature naive T cell that is actually going to deliver signal 1 to the mature naive T cell. Then what I want to talk about is co-stimulatory molecules. Okay, so this will involve then studying the T-cell receptor in a little bit more detail than we've studied before, or rather what's known as the T-cell receptor CD3 complex. So what I want to show you is the T-cell receptor CD3 complex. So I'm just going to draw out what we've seen of the T-cell receptor previously, and then I'm going to add on this molecule called, well, this protein complex called CD3. So T-cell receptors on the surface of mature naive T cells do not just exist in the form that I have shown you previously. They don't just exist as the alpha chain and the beta chain bound together on their own. Instead, what happens is they bind with a complex of 6 proteins known as CD3, and this T-cell receptor CD3 complex is then what's going to actually function in delivering signal 1 to the mature naive T cell when it actually binds to the MHC peptide fragment complex. Okay, right, so let me just draw out the T-cell receptor CD3 complex. So let's say that this is the cell membrane of our mature naive T cell, and let's say this is the extracellular fluid and this is the intracellular fluid. So I'm going to begin just by drawing out the alpha chain and the beta chain again, so this is good revision for us. What's that? Let me just get rid of that. Okay, so we'll start with the alpha chain firstly. So remember, both the alpha chain and beta chain consist of 2 important domains, the variable domain and the constant domain. And it's the variable domain that is different between the different T cell clones. So this is the variable domain of the alpha chain, and this is the constant domain of the alpha chain. And I'll just colour in our alpha chain in green here. Okay, so there's the variable domain and there's the constant domain. Then let's do the beta chain. So beta chain is very similar. It's also got 2 important domains, a variable domain which will be different between the different T cell clones, and a constant domain here, and then it's got its membrane spanning alpha helix and its C-terminus on the intracellular side of the membrane. So this is the variable beta domain, and this is the constant beta domain. And I'll just colour this in here in blue. Okay, right, so that's the T-cell receptor made up of the alpha chain and the beta chain. So that's the TCR portion of this complex. Now what we want to discuss is CD3. Now CD3 is a protein complex that consists of overall 6 different polypeptides. So I'll just call them 6 different proteins. Okay, now these 6 different proteins are also known as chains. So we stick to the chain notation. We've talked about the alpha chain, the beta chain. We're now going to call the 6 polypeptides that are going to be part of the CD3 complex chains as well. We're also going to stick to the Greek nomenclature. So we're going to have 1 gamma chain, 1 delta chain, 2 epsilon chains, and 2 zeta chains. So let me start by putting on the zeta chains. So the zeta chains are right in the middle of the T-cell receptor. So they're usually drawn in this sort of position here. Again, they're membrane spanning proteins, so I've just shown them spanning the membrane there. And they're going to be bound to one another very tightly, like so. And then the 2 of them, the dimer of zeta chains here, is then going to be bound to the T-cell receptor complex here. So these 2 proteins that I'm drawing here, these are representing the zeta chains of the CD3 complex. So I'll just colour in the zeta chains in red here, and you can see that I have just shown them having a membrane spanning region, and indeed they are integral membrane proteins like so. So those are the zeta chains of the CD3 complex. We're also going to have a gamma chain and a delta chain. So these are going to be bound on the sides of the T-cell receptor complex, like so. So here I'll show my gamma chain, and I'll put on the other side the delta chain here. So this is the gamma and delta chain. I'll just colour these in as well. So we'll have the gamma chain here in pink, and we'll have the delta chain in yellow. And then finally, we're also going to have 2 epsilon chains that are going to be part of the CD3 complex. And I'll draw these on either side like so. So we'll have an epsilon chain here. So this is an epsilon chain. And then we'll have another epsilon chain over here. So overall, those are the 6 proteins that are part of the CD3 complex, and they're all going to join on to the T-cell receptor complex to form the T-cell receptor CD3 complex, like so. So this is the way that the T-cell receptor actually exists in the surface of the membrane of the mature naive T-cells. It doesn't just exist as the alpha chain and beta chain. We've got these CD3 proteins bound with it. Okay, so what I now want to go through is the signaling cascade that is actually going to lead to signal 1 when this complex forms. Okay, so let me now draw this out again. Now I'm going to draw a simpler picture because that picture took me quite a while to draw. So I'm going to draw a simpler picture to represent this whilst we're looking at the signaling cascade. So all I'm now going to do is I'm going to show the alpha chain and the beta chain just as lines now. So here this will represent the alpha chain, this will represent the beta chain, these 2 proteins here will represent the zeta chains, which is this Greek letter. Oh, I should have actually introduced this Greek letter because you might not totally be used to this Greek letter. This is the Greek letter zeta. Okay, I hope you're familiar with delta, gamma, and epsilon, but you might not have seen that Greek letter before. That's the Greek letter zeta. Okay, so these are the zeta chains, and then on the side we'll have the gamma and delta chains, and then here are the epsilon chains, like so. So I'll just colour code it in again. So here is the alpha chain in green, here's the beta chain in blue. We'll go for the zeta chains next. So we've got the 2 zeta chains here in red. We'll have the delta chain again in yellow, the gamma chain in pink, and then finally the epsilon chains in orange. So here's my quicker picture for drawing the Tcell receptor CD3 complex. Okay, now let me draw on my MHC complex here. So here is my MHC complex, and again we're staying completely general. This is the same for both the cell-mediated pathway and the humeral pathway. So I'm not saying whether this is specifically MHC class 1 or MHC class 2. The pathway will be exactly the same for both of them. Okay, so I'll put my MHC protein complex here. So I'll just colour this in again in orange like so. And we'll have the peptide fragment, of course, in the peptide binding groove there. So this is either MHC class 1 or it's MHC class 2. And here is my peptide fragment there. Okay, right, so the Tcell receptor is bound there, and now, of course, what we need to add on is our co-receptor. It's either going to be CD8 or CD4, as we've just discussed. So I'm going to now draw on the CD8 here, like so. Okay, now what I want to do is discuss how this is going to activate an intracellular signaling cascade that is actually going to lead to signal 1 inside the mature naive T cell. So this is either CD8 or it's CD4. And again, I'll colour this in pink here. Okay, so this pathway is very similar to receptor tyrosine kinase pathways, but with a little difference, one little difference, which is that the tyrosine kinase isn't actually built into any of the receptors here. Instead, it's going to be attached on, it's going to be a cytoplasmic tyrosine kinase instead. But the principle is going to be very similar to receptor tyrosine kinase pathways. It's going to involve the phosphorylation of tyrosine residues on the intracellular aspects of proteins involved in the receptor complex. Okay, so let me now go through how this pathway is going to proceed. So when you form one of these complexes, the key thing that's happened here is that you've brought the co-receptor here, the CD8 or the CD4, close to the Tcell receptor CD3 complex. That's the key thing that this does. You bring these two very close together, and the co-receptor, the CD8 or the CD4, is going to bring with it a tyrosine kinase. So attached onto the cytoplasmic aspect of the co-receptor, and this is for both CD8 and CD4, there is what is known as a cytoplasmic tyrosine kinase, and this is just a free tyrosine kinase enzyme. It's an enzyme that floats around in the cytoplasm and which catalyzes the phosphorylation of tyrosine residues of proteins. So this is a cytoplasmic tyrosine kinase, and this one is specifically going to be attached to the cytoplasmic aspect of CD8 and CD4 molecules. And the name for this is LIC, or LCK. Okay, so this is the LCK cytoplasmic tyrosine kinase. So LIC is an example of a cytoplasmic tyrosine kinase. And I'm just going to colour LIC in here in orange. Okay, so we're going to see lots of different examples of cytoplasmic tyrosine kinases, and they all have names similar to this, 3-letter names. So this is LIC, our first example of a cytoplasmic tyrosine kinase. Right, so this is the key thing that happens. When we form this complex, it brings the LIC enzyme that's attached to the cytoplasmic aspect of the co-receptor very close to the cytoplasmic aspect of of all these proteins that are involved in the Tcell receptor CD3 complex. Now, a lot of these proteins, so for instance, all the proteins of the CD3 protein complex, have tyrosine residues on their cytoplasmic aspect which can be phosphorylated by the LCK or LIC enzyme. Okay, so let me draw this now. So I'll just show these as little lines. So these little lines that I'm now adding on to the cytoplasmic aspect of my CD3 proteins here, these are representing tyrosine residues that are in the cytoplasmic portion of the protein. And now that LIC has been brought so close to these tyrosine residues, what it's actually going to do is it's going to phosphorylate them. Okay, so these tyrosine residues are going to get phosphorylated, and the way I'm going to show this is I'm going to add a pink blob onto them. So this is representing the phosphate group that has been stuck onto those tyrosine residues. Okay, right, and now what's going to occur is that these phosphotyrosine residues, as they're now called, so once you've stuck a phosphate group onto a tyrosine residue, it's now referred to as a phosphotyrosine residue. These phosphotyrosine residues are now going to serve as docking sites, adhesion sites for another cytoplasmic tyrosine kinase, and this cytoplasmic tyrosine kinase is known as SYK. This is a slightly more famous one than LIK. So the next one that's going to come up is called SYK, and this actually I know what it stands for. This stands for spleen tyrosine kinase. So the S is for spleen, the Y is the single letter amino acid code for tyrosine, and then K is for kinase. So Y stands for tyrosine in the single letter amino acid code that's been agreed on by biochemists. Okay, so spleen tyrosine kinase is another example of a cytoplasmic tyrosine kinase. It's usually floating around in the cytoplasm of cells. However, once you've got these phosphotyrosine residues on the intracellular aspect of your CD3 proteins, the spleen tyrosine kinase, the SYK enzyme, it can come and bind to these phosphotyrosine residues like so. Okay, so here it is. The other key thing that I should mention about the spleen tyrosine kinase is that at the moment it's inactive. So when it's normally just floating around in the cytoplasm of the cell, it's not active. It's got a very low activity. It's not really going to actually phosphorylate any tyrosine residues on proteins. Okay, but what can now happen is when we get these phosphotyrosine residues appearing on the intracellular portions of the CD3 proteins, it can dock onto those phosphotyrosine residues. So here I've shown it coming and binding to one of the phosphotyrosine residues on, this is an epsilon chain of the CD3 protein complex. And now even that docking doesn't yet activate it. It's not activated yet. The next stage in its activation is that it actually has a tyrosine residue, a phosphorylation site, that if that phosphorylation site is phosphorylated, will activate the enzyme. So to activate the spleen tyrosine kinase, you need to phosphorylate it on a certain tyrosine. And can you guess what is going to perform that phosphorylation now? Well, it's going to be the LIK enzyme that's very close by. So what now happens is once you've recruited SYK enzymes to the CD3 protein complex like so, the LIK enzyme is going to then phosphorylate its special phosphorylation sites here. So I'll just show this like so. So here, there's the pink blob denoting the phosphate group on that tyrosine residue. And that now is going to lead to the activation of the spleen tyrosine kinase. So SYK is going to become activated. So I'll just draw sparks there. So it's now going to phosphorylate tyrosine residues on other proteins, and this enzyme is now going to cause signal 1. So this is the point where I'm going to stop the signaling pathway. We're going to stop at this point. This protein is going to phosphorylate other proteins, and the changes that that brings about is then going to cause signal 1. So we're going to summarize it as so. Once you've got SYK activated, that causes signal 1 to be delivered to the mature naive T cell. So just summarizing this pathway then. This is the pathway for the delivery of signal 1 of T cell activation to a mature naive T cell. When the antigen presenting cell arrives with its peptide fragment bound in its MHC class 1 or MHC class 2 molecule, it's going to bind to the T-cell receptor, presuming that we have found the correct T-cell clone. And what's then going to happen is the co-receptor is going to also come and bind to that MHC molecule. So, of course, if we're dealing with MHC class 1, the co-receptor will be CD8. If we're dealing with MHC class 2, the co-receptor will be CD4. The binding of the co-receptor brings the co-receptor extremely close to the Tcell receptor CD3 complex, and the co-receptor has bound to its cytoplasmic aspect the LIK enzyme, the LCK enzyme, and this enzyme is permanently activated. Okay? Now, once we've brought this Lick enzyme very close to the intracellular aspect of the CD3 proteins, it can phosphorylate tyrosine residues on these CD3 proteins. And these phosphotyrosine residues that are now appearing on the cytoplasmic aspect of the CD3 proteins, they're going to serve as docking sites for the SYK enzyme, the spleen tyrosine kinase, which is another example of a cytoplasmic tyrosine kinase. Now, the SYK enzyme is not normally activated, but what's going to happen now is we've brought it up, we've now got it joined onto this great big complex that we formed here, and now it's very close to the Lick enzyme. And in fact, to activate the SYK enzyme, you need to phosphorylate a certain tyrosine residue of that protein, and that's what the Lick enzyme is now going to do. It's going to phosphorylate this special tyrosine residue on the SYK enzyme and result in the activation of the SYK enzyme. The activated SYK enzyme is then going to phosphorylate loads of other protein targets, and that's going to deliver signal 1 to the mature naive T cell. Okay, so that's signal 1 of activation of T cells. However, in order for T cells to become activated, you need more than just signal 1. You need also co-stimulatory molecules. So this is the topic that I now want to move on to, and this is another way to prevent autoimmune responses from occurring. So co-stimulatory molecules is what I now want to talk about. So here's the story. If you just do what I've described to you so far, i.e. if the antigen presenting cell just forms this complex that I've shown you, it brings an MHC protein complex with a peptide fragment on it, and it finds a T cell clone with a correct T cell receptor to bind to that, and it delivers signal 1 to that T cell. Okay? That T cell will not be activated to divide and divide and divide. It will not be activated to start an adaptive immune response. Instead, what will happen is it will go into a phase known as anergy. Okay, so if you just get signal 1, if a T cell just receives signal 1, it goes into something known as anergy. Now, what is anergy? Anergy is not death, it doesn't die, but what happens is it can never now be activated. It goes into a quiescent behavioral state where it will never ever be activated, ever. Nothing that any antigen presenting cell does to it will ever manage to activate it. Okay? It will not divide, it will not take part in homeostatic proliferation, it will just live out its life quiescently. Once its lifespan is over, it will be removed from the bloodstream, and then that T cell clone will disappear because it hasn't taken part in homeostatic proliferation. And you might ask, well, why? Signal 1? Why should signal 1 do that? Well, if you just receive signal 1, it indicates that the peptide fragment that is trying to activate that T cell in the MHC complex, i.e. the peptide fragment that the antigen presenting cell is presenting to that T cell, is most likely a self-peptide fragment. What has to happen is you also have to receive signal 2, and this is the signal that tells you that the thing that induced signal 1 is probably a pathogenic antigen fragment rather than a self-peptide fragment. So this is another layer of protection against autoimmune responses. Okay, so we're now going to discuss co-stimulatory molecules. Okay, so in order then to actually get the mature naive T cell to become activated, it needs to receive not just signal 1, but also signal 2. And let me firstly describe what is actually going to lead to signal 2 being produced in our mature naive T cell, and then I'll explain what actually causes signal 2 and how this actually functions as a checkpoint against launching adaptive immune responses against self-peptide fragments. Okay, so I'm just going to draw out this picture again. So let's say that this is the cell membrane of our antigen presenting cell, so this is going to be our antigen presenting cell, and let's say that this is the cell membrane of our mature naive T cell, which will either be a CD4 positive or a CD8 positive mature naive T cell. And I'll just quickly redraw out the MHC complex here, binding with our T cell receptor, whoops, like so. And then here is the co-receptor here, and we know now that this will be delivering signal 1. Okay, and the peptide fragment will be here. So this is the Tcell receptor. I'll color code this in to make it look more interesting. So here is the Tcell receptor, once again colored in red. Here is the co-receptor, colored in pink. Okay, then we have our MHC class 1 or MHC class 2. Again, we're not specifying, everything's still general. We will have to specify soon, but at the moment the pathway is completely general for both cell-mediated and humoral adaptive immune response. And there's the peptide fragment. So this delivers signal 1, and we understand signal 1 now. Okay, what also needs to happen is a molecule on the antigen-presenting cell, known as a B7 molecule, needs to bind to a molecule on the surface of the mature naive T cell known as CD28. So there is a molecule on the surface of the mature naive T cell, a receptor on the surface of the mature naive T cell known as CD28. And it is essential that this receptor is also activated at the same time as you are delivering signal 1. If it's not, and you just receive signal 1, then the T cell is going to go into anergy. Okay, so this CD28, this is what delivers signal 2 here. And together, signal 1 and signal 2 will then lead to signal 3, which I'll describe in a moment, which will then cause full-on T-cell activation. So, what is the ligand then for CD28 on the surface of the T-cell? Well, it's a molecule that the antigen-presenting cell must put on its cell surface membrane if it's going to correctly activate the mature naive T cell, and this is known as a B7 molecule. And there are two different types of B7 molecule that can both perform the same function. One is called B7.1, the other is called B7.2. So nice and simple there. So here in yellow, these are representing the B7 molecules. Okay, so that's what has to happen in order for the T cell to actually be activated. It must receive both signal 1 and signal 2. So, what I now need to discuss is what on earth is the point of this? How does this actually function to protect us against autoimmune responses? When do antigen-presenting cells put B7 molecules on their surface? So the important thing to say is that antigen-presenting cells, macrophages and dendritic cells, do not have B7 expressed constitutively. They don't normally express B7. It's something that they only put on their surface if they have been activated in other means, i.e. if they've had pattern recognition receptors on their surface activated. So going back all the way to our discussion of the acute inflammatory response, remember, macrophages and dendritic cells had a huge number of pattern recognition receptors on their surface. So I'll just put a little discussion of this, a little reminder of this over here. So if this is our macrophage, which is our representative for the antigen-presenting cells, they have on their surface a huge range of pattern recognition receptors. Okay, and these pattern recognition receptors will be activated by pathogen-associated molecular patterns, molecules that give you a huge amount of indication that there is a pathogen present. And remember, when these pattern recognition receptors were activated on the surface of these sentinel cells, it caused the activation of the sentinel cell, and the sentinel cell started producing all of those pro-inflammatory mediators that we discussed. So interleukin 1, tumor necrosis factor alpha, interleukin 6, the prostaglandins. Okay, it started making all of those. Now here's the new bit of information. That's when they also start expressing B7. So macrophages and dendritic molecules will start expressing B7 proteins. And by the way, this is a protein. It's nothing to do with the vitamin B7, so don't get confused there. It's just the same name. So it's a protein, B7. Well, two different proteins, B7.1 and B7.2, which both perform the same role and are collectively known as B7 molecules. So that's when these macrophages and these dendritic cells are going to start expressing B7. So they're only going to be expressing B7 if they have seen pathogen-associated molecular patterns which have activated their pattern recognition receptors. So this, hopefully you will now agree, is going to function as a checkpoint. This is making sure that this antigen-presenting cell that is about to activate this T cell has actually come in contact with some pathogen. If it hasn't got B7 molecules on its surface, then that suggests that this antigen, well, this peptide fragment here that is in the MHC protein complex and is about to activate this T cell may well be a self-peptide fragment, because why have we not got B7 molecules on our surface if it has indeed come from a pathogen? Okay, so this is how this actually functions as a checkpoint. So just saying a little bit more on this, I want to introduce the concept of central tolerance and peripheral tolerance. So central tolerance and peripheral tolerance. So we have now seen lots of mechanisms to try and prevent autoimmune responses. Tolerance in the context of immunity and the immune system, or immunology, tolerance means not actually launching adaptive immune responses against self-peptides, so being tolerant to your self-peptides. So it's the word for avoiding autoimmune reactions. Now, what we have just seen is an example of a peripheral tolerance mechanism, but we've also seen loads of examples of central tolerance mechanisms. So all of those negative selection mechanisms that we've seen when T cells were being developed in the thymus gland, which were intended to destroy developing T cells that had T-cell receptors which could be activated by self-peptide fragments presented on MHC protein complexes. Those mechanisms are known as central tolerance, okay, because they're occurring in the development process for the lymphocytes. This mechanism is an example of a peripheral tolerance mechanism. It's a mechanism that exists to try and prevent autoimmune responses if central tolerance has for some reason failed to eradicate this T cell. So if this T cell does have a T cell receptor that can be activated by a self-peptide fragment on an MHC protein complex, then, i.e. that central tolerance has failed, then we have another mechanism, another layer of protection, and this is now what is known as a peripheral tolerance mechanism. So this is a major example of our peripheral tolerance mechanisms, that if an antigen-presenting cell attempts to activate a T cell when it doesn't have B7 molecules on its surface, then it will only deliver signal 1 to that T cell, and the T cell will go into an anergic state. It will go into anergy, and it will now never be activated. It can never be activated. It doesn't die, it lives quiescently, it doesn't divide, but it will never be activated, and eventually it will just be removed from the bloodstream because it will die, and then that T cell clone will die out in the body, and that's a very strong protective mechanism to try and prevent autoimmune adaptive immune responses being initiated. Okay, so that's an example of peripheral tolerance. Now let's go back to the story of we've actually got a pathogen's antigen fragment here, and this is not an autoimmune response, so the antigen presenting cell should have B7 on its surface because it has seen a pathogen, and therefore we will be giving our mature naive T cell both signal 1 and signal 2. What's now going to happen? Well, what's now going to happen is signal 1 and signal 2 are going to result in the T cell receiving signal 3. So signal 1 and signal 2 together, they cause changes in gene expression in the T cell, and these changes in gene expression are going to allow a form of autocrine signaling, which means when a cell releases the molecule that's going to actually stimulate a receptor on itself. So auto means self, so autocrine signaling is where you signal to yourself, and I'll give you an example of this in just a moment. So you're going to produce proteins that are going to allow autocrine signaling, and this is then going to deliver signal 3 to the T cell. So we're going to change gene expression, and two of the major genes that we're going to start expressing are the gene for interleukin 2, IL-2, and also the gene for the interleukin 2 receptor alpha component. Okay, so let me firstly talk about the interleukin 2 receptor alpha component. So mature naive T cells, they usually have two of the components of the interleukin 2 receptor on their surface. So the interleukin 2 receptor It's made up of 3 separate proteins, the interleukin-2 receptor alpha component, and then the interleukin-2 receptor beta component, and the interleukin-2 receptor gamma component, which I'm drawing here. So this is the interleukin-2 receptor beta component, and I'll just colour this in. So this is supposed to represent the cell membrane of our mature naive T cell here. And this is the interleukin-2 receptor beta component here in orange. And then bound to it is the interleukin-2 receptor gamma component in green. So this is interleukin-2 receptor gamma component. So normally mature naive T cells, they only have interleukin-2 receptor beta components and interleukin-2 receptor gamma components actually expressed. They don't make interleukin-2 receptor alpha components. And this means that the interleukin-2 receptors are only partly assembled on the surface of the mature naive T cell. They're missing a crucial bit, which is the interleukin-2 receptor alpha component. So this does not work. This is not a viable receptor for interleukin-2. This will be doing nothing. So, you can see that what this T cell is now going to do once it's received signal 1 and signal 2 is it's going to produce the final component of the interleukin-2 receptor, and it's also going to start producing the ligand for the interleukin-2 receptor, which is of course the interleukin-2. So you start producing interleukin-2 receptor alpha components. Those will go to the cell membrane, and you'll now end up with a complete interleukin-2 receptor, complete interleukin-2 receptor proteins on your cell surface. So the T cell is now going to have complete interleukin-2 receptors on its surface. The interleukin-2 molecules that you produce will be secreted from the cell. So here they go. So they're going to go into the extracellular fluid, and then they're going to act on the receptors on the same cell that secreted them. This is why this is called autocrine signaling. So when a cell releases a molecule that's then going to work on receptors that are on its own cell membrane, that's called autocrine signaling, and we've got a lovely example of autocrine signaling here. So here's the interleukin-2 molecule now binding to the interleukin-2 receptor on the mature naive T cell, like so. Once this has happened, the interleukin-2 receptor complex will then activate a signaling cascade inside the T cell, which will then cause signal 3. So this intracellular signaling cascade is known as signal 3 inside the T cell. So signal 3 of T cell activation. And we will see the effect of signal 3 of T cell activation in the next video. For now, let me just summarize what has happened. So, our antigen-presenting cell has brought in this peptide fragment from a pathogen here, okay? And because it's seen a pathogen, it's also put in B7 molecules onto its cell surface membrane here. The MHC peptide fragment complex has managed to find a T cell that has the correct T cell receptor that can actually bind to it. This T cell receptor will bind there. The co-receptor will also bind, and if this MHC was MHC class 1, then the co-receptor will be CD8, and the T cell will be a CD8 positive T cell and will be involved in the cell-mediated adaptive immune response. If this MHC protein complex was MHC class 2, then the co-receptor will be CD4, and this T cell will be a helper T cell involved in the humoral adaptive immune response. And we'll see the difference later on. So, together the T cell receptor and the CD8 or CD4 will initiate signal 1 inside the T cell. Because you've also put the B7 molecules on your surface, you will also get those binding to CD8 proteins on the surface of the mature naive T cell. And I want to stress, although I've just shown one of these here, you'll form lots of these complexes in the junction between the antigen-presenting cell and the T cell. And these will initiate signal 2 inside the T cell. Signal 1 and signal 2 both received together, they have to be received together in order for this to happen, will cause a change in gene expression within the T cell, which is in favour of the T cell becoming activated. And this is going to result in the T cell producing both interleukin-2 and interleukin-2 receptor alpha component. The interleukin-2 receptor alpha component will go to the cell surface membrane and bind with interleukin-2 receptor beta and interleukin-2 receptor gamma to form a complete interleukin-2 receptor on the surface of the T cell. something that the T-cell never had previously. It did not have complete interleukin-2 receptors. The interleukin-2 that the T-cell has produced will be secreted from the T-cell and will then act on these complete interleukin-2 receptors and will activate an intracellular signaling cascade that delivers signal free to the T-cell, and that's actually going to activate the T-cell. As we'll see in the next video, the response of the T-cell is now to divide and divide and divide and divide and produce loads of identical T-cells with an identical T-cell receptor, so-called T-cell clonal expansion, because we are expanding that T-cell clone that has a T-cell receptor against this antigen fragment from our pathogen. But we'll look at that in the next video.